Hi everyone, this is Jason, producer of Data and Biotech. Before we get started, I wanted to let you know about our latest white paper. It's a comprehensive guide to implementing machine learning models in biotech manufacturing. It's a complete overview of all the potential problems of ML adoption and, more importantly, how to solve them. To download it, simply visit connect.cordine.com/biotech-ml. We've also dropped the link in the show notes of this episode. Okay, let's get into it. Welcome to Data in Biotech, a podcast from Cordine where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. In this week's episode, we speak to Timothy Jenkins, Head of Data Science and Associate Professor at DTU Bioengineering. Tim shares his journey from studying zoology and venomous snakes to leading a research group focused on AI-guided drug discovery, studying the evolution of protein structure prediction, spearheading collaborative approaches in protein design, and managing the technical overview of antivenom discovery. He also impacts a very exciting case study on the use of AI in developing solutions for neglected diseases, particularly focusing on antivenom for snake bites, and finally shares his take on the integration of quantum computing with protein design. Here we go.
Tim Jenkins, welcome to the Data and Biotech podcast.
Thanks for having me. Excited to be here.
Awesome. Well, just to kick us off, can you provide us a brief introduction to your background and what brought you here?
I can make it a long or a short story. I'll give you a short one and where we can dive deeper. But yeah, my name's Tim. I'm an associate professor and head of data science at DTU, the Technical University of Denmark, just outside Copenhagen. And I run a small research group that is focused on AI-guided drug discovery and development.
Awesome. And what got you into that field?
Well, it's actually quite a wound path. I started up with zoology in Australia, was working with venomous snakes, and then I got increasingly interested in the venoms themselves rather than just the animals producing them. And that led me more and more down the molecular route and then the therapeutic route. And then during my PhD at the University of Cambridge, I realized that I also really like computational approaches a lot more than wet lab-based approaches, at least for myself. And I started venturing down that road and started looking more into the other end of the spectrum of how do we actually make therapeutics and how can we make them fast? And AI seemed like a smart way to go. So that's how I ended up where I am now.
Yeah, it's really interesting that you've been thinking about or working on snake venom for a long time. So, what made you passionate about exploring things related to snake venom?
Travel. I think that is probably the single best explanation for that. I traveled a lot when I was a child. When I was 4 years, I was in Indonesia, and that was the first time I got to hold a snake. It was a guy who had this really cool little ice cream truck, but instead of ice cream, he had snakes. I guess most kids would have been disappointed. I was thrilled, and I got to hold it there, and I was in love ever since. And then when I was 6 years old, I went to Australia and told my mum that I would study venomous snakes in Australia when I grew up. And at the time, she laughed at me and tapped me on the back. I was like, sure, kid. And yeah, 12 years later, I could tell my mum that I told her so, and she couldn't really complain that I was moving to Australia. So yeah.
Yeah, that's an amazing story. So can you talk to us about DTU Bioengineering and, the organization's mission and, what challenges you work on over there?
DTU Bioengineering is one of the many departments that we have at the university. And it's got several hundred employees. It's very much focused on biotechnology and biomedicine. And we've got a very diverse range of groups. Most of them are very wet lab focused, and some of them are very fundamentally interested. But it is a technical university, so we are training engineers. So a lot of them are very solution focused on developing next generation cell therapies or looking at how we can create bioindustrial enzymes and a lot of different areas that we're covering. And obviously, increasingly also focusing on driving data science, data literacy, data access. And that's also my role as head of data science to kind of at least pioneer the way and maybe even show the way to some extent and help lift up the whole department along those lines alongside my brilliant team in the data science hub there.
Awesome. Well, we at Data and Biotech are very excited to be a part of that. So, I know you've got some exciting projects going on at DTU, and probably the one that sticks out most is the soon-to-be released article in Nature about de novo protein design for neutralizing snake venom. Could you give me some background on the project and, the work you've done around creating binding proteins for snake toxins?
Yeah, I'm very biased on this, so take everything I say with a pinch of salt, but I'm very excited. I think it's a really, really interesting step forward in the path of the therapeutic discovery. And the way this whole thing started was that I saw this preprint at the time come out in late 2022 by David Baker's group. David Baker might ring a bell at this stage. He received the Nobel Prize in chemistry just last week. At the time, he was already quite popular and had put out a lot of Nature and Science papers and great science and tools. And he put out this paper that was very eye-catching to me because it really promised that protein design had now matured to a stage where it wasn't just cool, but actually useful. And I decided to reach out, thanks to also some good pushing from a close colleague of mine at MIT. And sent an email just before Christmas saying, hey, do you like snakes? And luckily enough, snakes have a way of getting people excited, and David got us started on this project. And it was very fortunate that at the same time, a graduate student in his group from Mexico, Susana, who's also the first author on this paper, had also started thinking about snakebite and was excited. And so the dots were connected and the project was started, which was very fortunate.
Before we dive into sort of the nuts and bolts of what you did in the paper and how you came to the conclusions that you did, can you give us a history of the effort from, the preprint you saw by David Baker all the way up to today? Because I know that this paper that you're just now releasing is sort of the latest milestone in a long line of milestones that you've achieved along the way.
Yeah, and I wish I could take the claim to fame for many of them, but I definitely cannot. David, however, can, hence he got the Nobel Prize and not me. Very deservedly so. So I think the biggest, push forward in my particular field here has been the computational solving of protein structures. So proteins, those molecules of life, they are the antibodies in our system defining us from pathogens. They mediate the communication between cells. They're essential to a lot of aspects in all living things. And one of the biggest problems in this space is understanding how these molecules form a 3D structure, because usually when you look at proteins, they are made out of amino acids, which can be seen as letters, but they don't exist as these strings of letters. They exist as three-dimensional molecules, and resolving this experimentally is extremely painful and time-consuming and not very high throughput at all. To give you an example, understanding, for instance, from a single snake toxin, and snakes can have up to 100 different toxins. A single toxin structure could take you a year to understand what the protein actually looks like in three-dimensional space. And so David Baker's group academically had pushed that field forward quite substantially with developing software solutions such as Rosetta, which try to rationally predict how these proteins would fold. But the field as a whole didn't really get close to solving it until AI engineers came into this whole field and developed something known as AlphaFold, which made a big splash in the community because they started applying the latest machine learning models that were focused on language, and biology in a way is a language. It's letters and sequences and words, right? So they applied the same rationale to protein structure prediction, really revolutionized our possibility to identify structures. And that is also why Dennis Hassabis and John Jumper got the Nobel Prize as well, alongside David Baker, for protein structure prediction, because they, in the company called DeepMind under Google, developed AlphaFold and solved this critical problem. And then David Baker's group came in and said, hey, okay, that's great, but the next step is really generating new things. How do we generate new structures from scratch? And people had been trying to do this rationally, also with machine learning, but the success rate was incredibly low. You would have to test 100,000 different predicted therapeutics, and maybe one of them would be okay. But that was awful because it would cost a lot of money, would take a lot of time. And we had other technologies that were very much wet lab-based molecular biology technologies that were better. They were faster, more cost effective. What was really shown in this paper was that the success rate went from 0.1% to up to 30%, which might not sound like a lot, but was completely transformative in this field, because now you could take 90 different variants into the lab and 30 of them would be decent leads that you could characterize further. And that was really what caught my eye. They did this for 6 different biological targets. So it was seemingly robust, but I had to see it for my own eyes and try it out myself before I really believed it, because scientists are skeptical. But I was convinced, and I am very convinced that this technology works and is really transforming the field that I'm part of.
Yeah, so you believed in it and you needed to try it out for yourself. Can you walk us through sort of the journey of trying it out for yourself and how you went from sort of reading about the new AI technologies that have become available for the structure prediction and for the design of proteins to actually designing your own proteins for antivenoms?
Yeah, so I mean, we've been working a lot already with AlphaFold and protein structure prediction. And I think a key part in our field and in general is open science, right? There's a lot of people that are publishing amazing models, but they're closing the code, they're closing access, or they're not making it available for commercial use, which complicates broad adoption. But some of these tools have become very broadly adoptable. I think AlphaFold 2 was a great example. And the same thing happened with David Baker's tools that has had a very open access attitude, and I think played a major role in him getting the Nobel Prize, because it opened up the field for so many people to do amazing things. And so we've been playing around with some of the tools that have come out, and we are starting to play around with the new protein design tool. But what was beautiful is actually seeing the experts use it and learning how they evaluate outcomes, how they work with it. And so in this particular project, we did not really do in my group a lot of the protein design. That was really led by Susanna at the Baker Lab, but we were part of the design strategy, understanding how do we need to target these toxins to actually make effective therapeutics. And so learning by collaborating, I think, is always the best way to work with the experts and then onboard it. And now we're using this technology, because it's open access, across a lot of different projects for bioindustrials or other therapeutic applications. And yeah, it's pretty amazing. It's quite user-friendly, I have to say.
I'd just love to go through sort of a technical overview of the approach that you and your partners used to discover the de novo antivenoms. So that from sourcing the biological data to, training and conditioning, parameterizing the models, the different, like the ways that you brought sort of like experimentation or, external sources of measurement into the pipeline. So if we could just think about it from an end-to-end perspective, how the process worked, that'd be really helpful.
Of course. So I think that the first important thing here is we didn't build a tailor-made model for this particular application, which is also the really transformative part of this technology. You can throw it at any biological problem that has a protein in it and use it, which is brilliant. No fine-tuning, no nothing. It's just a generally applicable tool. And I can talk a bit about how that tool was built, because it's, I think, quite interesting. A lot of the things that are successful right now in biotech and biopharma are all derived from either large language models or image AI. And in this particular case, we've got AlphaFold, which we use quite heavily, and I'll get into a bit more details later. But that was built on large language models and large language model architecture. But then the generative design really lent or borrowed ideas from things like denoising diffusion that are big in image AI, where we've now all come to see and love the weird and quirky images that Midjourney or DALL-E 2 spit out when you say, I want a monkey riding a bicycle on the surface of the moon, right? That same type of approach really was applied to the protein space. And the best way to explain that is that with proteins, luckily enough, over years and years and years and hard work, scientists have actually generated a lot of protein structures. And it's probably one of the nicest data sets that we have in biology are all of these protein structures that have been deposited under very rigorous criteria on something known as the protein database. So there's actually a large database, which I also have to say was one of the main reasons why AlphaFold could be so successful. It was the work of many scientists over many years that had established the foundation, the training data to build AlphaFold and make it so good. Now, traditionally, when we're thinking about protein and protein structures, we try and tell the model, here's a protein, it does X, Y, Z, learn. And we do this over and over again. The way we did with images, right? If we say we want a model that can identify a cat, we give it 10,000 or 10 million images of cats, label them as cats, and then we give it 10 million other images and say not cat. Then it'll be able to identify a new cat or not. And the main thing that these models couldn't do is generate a cat. If you asked machine learning models 5 years ago even to generate an image of a cat, it was a nightmarish creature that would haunt you. Now it's photorealistic. You can have the right lighting. You can even decide what camera it should use for taking the image. So, and the reason why this happened and worked is this denoising diffusion, where you can introduce what is known as this Gaussian noise, where you iteratively noise your image, so randomize the pixels in your image until it looks like your old TV screen from the 60s, 70s. And you use each of these iterative steps to train your model to then learn not just what is a cat, but how do I generate a cat from complete noise to something. And you can do the same thing with protein structures, where you, instead of just train on the whole structure, you take that structure and iteratively noise and randomize the location of each of the components of the structure, each of the amino acids. And then you've got all of these steps, you throw this as your model, and suddenly it starts learning how to generate a protein from scratch. And this on the surface level is very much what they did in this particular paper, using the same type of database and just iteratively noising and using this for training. And that's really pushed the accuracy of this generative design quite far.
Setting aside all the work that you did up front to sort of understand the toxins that were there inside of these snake venoms, you've got this goal of designing an antivenom that, neutralizes as many of these toxins as possible. I believe we're talking about RF diffusion as sort of the example of the diffusion model that, generates these, protein structures. Can you walk me through how you map from the toxins whose structures and then condition the model, the diffusion model, to understand how to design proteins that are, most likely to do the job you want them to do when you put them up against these snake venom toxins?
Yeah, so essentially you can not just train the model on individual proteins, you can also train it on protein complexes. So examples of where protein interact. And in the end, that is the aim of what we're trying to do. We take a toxin of interest and we say we want something that sticks to this toxin. And ideally it sticks in a position that prevents that toxin then from interacting with a receptor. So in the case that we had here, we had neurotoxins. So these toxins target your nervous systems, they paralyze you, they shut down your breathing and your heart, really nasty things. And we know they've got these 3 fingers, they're called 3-finger toxins. And if we manage to stick something to the 3 fingers, then we know this is not going to do the job that it has to be toxic. So what you can do with the model is it's learned how proteins like to be close to each other. It has learned how these proteins are going to likely interact. And on a biophysical level, it's very much what type of charges do you have on the surface, what type of bonds are being formed with different types of amino acids. And so what you can do now is just take the structure of your target, in our case, a neurotoxin structure, and tell the model, generate something that will have a beneficial shape to the target protein that you're looking at. So it creates basically the backbone, the hollow shell that perfectly matches your target, using, again, generative design by saying, here, this is the target, make something of this size with this type of properties to my target. And then you can use a second machine learning algorithm, a message passing neural network called protein MPNN, that does something known as inverse folding, where it then suggests a sequence, so the letters that you need, to fold into this backbone. So it's kind of the inverse AlphaFold, where you have a structure and you want the sequence. And with these 2 tools, you then get your complete protein, your therapeutic protein, and then you can throw it into AlphaFold and use AlphaFold as a way of understanding if what you designed makes any sense or not. And there's different types of metrics. There's model confidence, the intrinsic model confidence from AlphaFold, and a lot of other things that you can look at to say, okay, is this likely going to work if we take it into the lab? And the combination of this design with the filtering has really pushed the success rates of what you take into the lab up a lot.
So I'm imagining that you go through this sort of process iteratively where you're not exactly sure what the specific binding targets are or the specific locations of the binding targets, or maybe you are. I'm just interested in how you refine that feedback loop that you've described in order to get closer to the thing that you're trying to design.
Yeah, so it's a great question. Like, one of the powers of this technology is that you actually have a very good understanding on where your binder will bind, which is one of the biggest advantages, I'd say, over traditional antibody discovery, where you're basically trying to fish things out with a magnet out of a soup. And then it sticks to the magnet, but I don't know where exactly, just somewhere sticking to the magnet. In this case, we're designing. And what we found in many cases, in this paper we've shown it, but I've also seen this in other projects, and David's also in many other projects. If you have something that binds, we know exactly where it binds. We have complete epitope-paratope control, and you can even try to condition the model to target specific areas that you're interested in by saying, hey, focus your design strategy on this area. So you give so-called hotspots on the target. Now, after the evaluation AlphaFold, what you can do is, if you're lucky, you already have designs that have the right scores, the right metrics that you're interested in, and you can go and take it straight to the lab, which we actually did in some of the cases. But by and large, what we're doing primarily in my group now is actually going through as much computational refinement as possible, because it saves money, it's cheaper and faster to do than doing it in the lab. And so what you can do is then take what is known as partial diffusion. So it's a bit like computational affinity maturation, so binding strength maturation, where you say, here's a good starting point. We know this binder looks good, but we think it can be even better. And instead of starting from complete scratch, it starts with that and then tries to change bits and bobs. And you can do this both on the backbone level, so on the structure, and then you can also do you can redo the message passing neural network, the protein MPNN, to suggest a bunch of different sequences, then evaluate all of that again in AlphaFold. And you can keep doing this as much as you want, really, until you're happy. And sometimes this is very easy to do. You get very good scores very quickly, and sometimes not.
Right. So you can, once you have that feedback loop established, you can sort of go down as many paths as you feel you need to go down. And, when you reach a point where you're no longer improving, but you're not where you need to be, then you need to go back to some earlier starting point and start again and see if you can, and sort of increase the diversity of the proteins that you're designing to see if there are other paths that sort of lead you to better endpoints. Am I thinking about that right?
Yeah, exactly. Diversity is actually a great thing to highlight here, because that is part of the strategy, is that there might be fundamentally something that the models get wrong, some assumptions that are ingrained in the models due to the training data and just information they see, they might just be inherently wrong. So one of the things you're trying to do is not just optimize your metrics, but also de-risk. And you de-risk by having different proteins that are all predicted to be good. So there's these two things that you're balancing, and it's a bit of voodoo at this stage. Nobody really knows where the sweet spot lies, and it depends a bit on the target, but there's definitely a lot of opinions on where the sweet spot could be. And a lot of discussions with my students are exactly on this topic. How do you weight these two things properly? And it's really interesting, because the metrics that you have are also not linearly correlated with success. We have a cutoff where we know under this, there's a good chance that they'll work. And then if we drive these scores even further, there's sometimes an increased chance they will work, sometimes not. And it's still a bit of a black box that is really exciting to explore now.
Yeah. So, I mean, that brings up the clear next question, which is when do you know that it's time to go into the lab with the sequence or sequences that you've designed in order to, try to validate that what you've designed is actually doing what you expect it to do?
Yeah. So very simply, we've got some very standard, I'd say, computational metrics where you say, if we're under this, it's worth taking in. And then, as you also mentioned, we try and improve those as much as possible. But when we see no further improvement, but we have something that's underneath those minimum thresholds, then we say, okay, let's go, let's take it to the lab. And the beauty of that is you can actually do a lot of iterations, again, computationally before you do that quite quickly. So you can go into the lab within a week of starting to design. Often it takes a bit longer, but can go very quick.
So you mentioned that, with snake toxins, you're dealing with, 100 different toxins. And so, one of the challenges here is that there's just different breeds of snake. There's the three-finger toxins that you mentioned. There's, SVMPs. There's just different toxins that you need to work with biologically. So I'm just curious, is what you designed covering all of those toxins or a portion of those toxins? Or, what is the journey like from here in order to get to an antivenom that sort of, takes care of all the entire scope of what you need to take care of in order to be sort of like a comprehensive antivenom?
Yeah, I wish I could tell you we've solved it. We've neutralized all snake venom toxins. Now, if you go to Sub-Saharan Africa, just give me a call and I'll give you some of our AI binders. If that were the case, maybe I would get the Nobel Prize in Medicine next year. Sadly, we're very far away from that. What we've done now is shown in a couple of the most lethal and problematic toxin families that the concept works. It's really a proof of concept study showing this actually works in neutralizing some of the most lethal compounds that are present in snake venom. But to really make a product, we've got work at hand, and that is what we are working on now, is really focusing in on a given product, solving a problem for a specific region, because that is what we want to see. We want to see AI tech solve neglected diseases and really unlock a lot of these different neglected, especially tropical diseases, and create cheap solutions for those people most in need. So but it is a long road ahead, and I don't want to oversell what we've got here.
No, not at all. But I think that what I'd like to understand is, what does the journey look like from here, from your perspective? So what are the next steps that you need to take in order to get to the product that you just described?
Yeah, no, happily. So, what we've really shown here is that these AI designed binders, the first time, as far as I'm aware, these diffusion-based binders work in living creatures. They don't just work a bit, they work excellently. We saw full neutralization across different settings, and we did this in a unprecedented amount of time, where we've been doing this with traditional antibody technologies in some of these toxin cases, and we've been working on this for 2 years in the lab. We had binders that were better in a matter of a month and a half in this case. So really speeding up the process. So the next step really is, one, focusing on broad specificity. So having single binding proteins, single therapeutics that are able to neutralize a whole subgroup of these toxins. And then doing this for more different toxin families, so we can really make this anti-toxin cocktail of components that can go in. Then obviously safety is something that we need to look into. A lot of people are worried about AI proteins or AI products going into humans, and there should be. It's something we should be at least looking into quite carefully, because these are foreign products that our immune systems has never seen. We were very happy to see that this wasn't an issue in the mice that we investigated in the neurotoxicity. There was no obvious off-target effects, immune reactions. But having said that, you will need to evaluate that. So we will do that. And then a lot of it is about, how do you scale up this production? Luckily, these binders express extremely well. You can make very, very large amounts of these on an industrial scale, at least based on the initial indications that we've seen. And we will need to investigate production strategy, formulation strategy, plan the clinical trials, which is a nightmare when you're talking about acute diseases such as snakebite, recruiting patients, standardizing. So there's a lot of things, and we're also in discussions with a lot of different partners who are experts in these areas, in planning clinical trials, in pushing drugs to neglected diseases. So it's going to be needing a large collaborative effort, and also a decent chunk of funding to back it up.
Yeah. And you mentioned that the protein that you've designed already, you're sort of relatively confident that, it can reach industrial scale at a reasonable degree of cost. Can you talk a little bit about, what leads you to those conclusions, or how this method of designing a therapeutic, enables that?
Yeah. So one of the things that is really nice is that all of these models are trained on proteins from a database, usually that have been crystallized. To crystallize a protein, you need a lot of it. It needs to express well. It needs to be stable. It needs to, by and large, be a well-behaved protein. So these models only really know what well-behaved proteins look like. Again, a bit of an exaggeration, but they definitely have learned more about that. And so intrinsically, they seem to be very good at creating well-behaved proteins. And across many different examples now, we've seen that, for instance, the thermostability of some of our binders is up to 95 degrees Celsius. Now, you might say 95 degrees Celsius, bit of an overkill. My bench is not so hot. But having very thermostable proteins and therapeutics is very relevant if we're talking about tropical countries where antibodies often aggregate. These proteins are very stable. Now, why do we think that they will also work well in industrial scale? Well, they express well in laboratory setting, also in production relevant hosts. So many academics say, my thing expresses well on my lab bench, so it will be great in industry. And it's not the same thing. There's very different types of systems and considerations. The beauty here is at the center of antibody technologies that I'm actually located at here at DTU. We've got a lot of experience there, and we work a lot with biopharma and bioindustrials where they understand how to scale up these proteins. And just looking at the way that the stability of them, the lack of aggregation, the way they behave in general in different systems in yeast, which is a major production organism now, we have high confidence that getting high yield production is very likely with these proteins.
That makes a lot of sense. So, I want to zoom out a little bit. So this de novo protein design for drug development project that you've gone through, it seems like it's a microcosm or an example of drug development processes writ large. In what ways is that true? And in what ways do you think that might not be true in the future?
I can definitely answer the first, but I think it is true in most cases. Again, biased opinion here, but you will often need to get a good therapeutic starting point to your target of interest. It doesn't matter if it's a snake toxin, a viral protein, a cancer epitope, or something related to autoimmune disease. It doesn't matter. As long as you're talking about a protein target and you need a protein to manipulate it, this is a very applicable process. You can go through the same steps, use the same tools. Again, thanks to them being commercially available, it doesn't even limit big companies from doing so. I think we'll probably see, there's always a time delay with novel tech being adopted by large pharma or large industry because there's a risk associated to it. It really needs to prove itself academically, and I'm doing what I can on my end to do that, because I really think if large industry starts picking up on this, and they are, we can really speed up also therapeutic development across many, many other diseases that really require treatment. And you will always go through very similar steps in terms of making sure your thing binds well, making sure it's a great protein that expresses well, is stable, it's developable. All of these same things apply to big pharma.
What do you think made antivenom development sort of a good starting place for this kind of process?
I think there's many different reasons. Obviously, it's an area I know very well, so it's always good to work in a field that you understand fully. It also is one of the areas that has lacked the most in terms of technological innovation because it's been severely underfunded. There's an example that is funny but really sad if you think about it, that up until a few years ago, more money had been spent on making the movie called Snakes on a Plane, which is obviously a masterpiece, but more money had been spent on that than actually spent on research on developing next generation solutions for snakebite. And if we look at that currently, if you were to go to Tanzania on a beautiful safari and are unfortunate enough to be bitten by a cobra bear, you might be lucky and they might have antivenom. And they give you that antivenom. That antivenom comes basically from horse blood. You've injected a horse with venom. You've waited a year for the horse's immune system to hopefully make some good therapeutic product. You then take the plasma from the horse. Don't worry, horse is fine. But you take the plasma, you purify it, and then you inject it into a human. So the technology there is very, very old school. It's still saving lives and it's essential. Trust me, if I got bitten, I want this in my bloodstream. But it's an area that really could use from novel tech. And that's what we've also been trying by and large at the center that I'm part of, where we're doing a lot of antibody discovery that is very successful and has shown very big promise in terms of actually providing a treatment for snakebite. But we're also looking at novel technologies such as this AI binder design here, because we really think it has the biggest potential to make an impact in neglected diseases or orphan diseases that are often overlooked by big pharma or big companies just because they have a responsibility to shareholders.
Yeah, that makes a lot of sense. So, looking back on this journey that you've been on, if you could give yourself some advice at the start of this research effort and, the beginning of design process to try to come up with de novo protein design for antivenom, what would you tell yourself, or what advice would you give yourself to, I don't know, avoid pitfalls, avoid wasted effort, conserve resources, something along those lines?
Yeah, I think it's a great question. It's a question I always ask my students at the end of their projects to get them to reflect, and find you get very interesting answers there. This is a tough one, and I am very happy to say it is a tough one because things went extremely well and extremely fast. So this is probably one of the most efficient scientific projects I've been part of. And that's also a big credit to Susanna, the first author, and also Melissa, who was the second author on the paper, did a lot of the work here. And I have to say, probably the best advice I could have given myself is actually be more ambitious, believe more in the technology, think more right from the get-go, like this is going to work, how do we make the product, rather than just see if the tech works. But having said that, I'm very happy we did it the way we did, and I couldn't be happier with the outcome. So yeah, not too many regrets to look back on.
For sure. Your paper mentions the potential of these methods to democratize therapeutic discovery. So I'm just interested in your thoughts on, how do you think this method helps to democratize therapeutic discovery?
The really cool thing here is that theoretically you can run the same tools that we used on your laptop. It will take a while. It will run hot, so don't keep it on your lap. But theoretically, that's possible. If you have a bit more luck, a bit more access to some slightly decent graphics cards, you can speed up the process quite a lot. That is very different to if I told you, make me an antibody in the lab versus make me a de novo binder with AI. If I ask you to do this in the lab, you need lab space, you need a lot of reagents, you need a lot of equipment, you need an antibody library to start with. There's a lot of startup costs you might not have in all areas across this globe, whereas having an internet connection, access to a decent computer is becoming increasingly prevalent, even in areas that are historically less economically strong. So I think that is a big change of mindset. And we've, for instance, in a slightly different project where we also developed our own AI model for sequencing actually snake toxins to identify our targets. We ran the largest hackathon in Africa with representatives from all over Africa. Nearly every African country participated. And there we could give access to the tool by them just having a laptop and an internet connection. They can be part of solving problems that are most relevant in their regions. I am not waking up and I'm worried about getting bitten by a snake in Denmark. If I do, that will be very, very unlucky. That's 2 people a year. But the people that are facing some of these diseases, why should they not be part of the solution? Why should they not figure out what is the best way to go forward? Because they understand the problem the best. And tools like this, and I think in general, having the digital age address some of these problems is a great way to unlocking a new talent pool that so far has had limited access to being part of the solution.
That makes a lot of sense. So if you were starting a new therapeutic design problem tomorrow, what would your first steps be? Where would you start?
I've got probably 8 running in parallel right now on very different target space in different areas. So, I mean, the best thing to do is think about a problem that currently is either got a poor solution or no solution. Ideally, something that you also personally care about because that really drives you. Understand the mechanism of that problem. Make sure that you have a problem that is solvable by a protein binder. And then get your structure. We've seen that even with an AlphaFold predicted structure, you don't need an extra crystal. It works. So you can have a target that is only predicted already, and then just start designing. And I think that's really the best advice I can give to people, especially young generation out there that wants to get into AI drug discovery. These tools are accessible. Try them out. Reach out to people who are in the areas that you're interested in. And just contact them, say, hey, I've played around with this tool. I think the area you're working on is great. There's going to be a lot of professors out there that have no clue on how to use these tools. If you come in and say, I love the biology you're doing, I've got a way to give you some cool reagents, cool diagnostics, cool therapeutics, I'm pretty sure they'll be excited to hear from you. So that'd be my best advice going outside.
Can you give us some intuition about where the de novo protein design space can be applied in terms of therapeutic development, and where you think the disease mechanism would not lend itself to this kind of solution?
In a way, you can look at it exactly like an antibody. It's a mini antibody. So anything that an antibody can do, these things can do. There are certain considerations, like since these generate the proteins, you can decide how big they are, but they tend to be quite small proteins. They don't stick around in the body so long. There's a way of engineering your way out, but that makes them intrinsically a lot more interesting for acute diseases, acute conditions such as snakebite. Doesn't mean you can't use them for chronic diseases, but you have to engineer your way out of that. And it's very doable. But at least if you had to choose between an acute and a chronic disease, I would probably go for acute. Because there also the risk of immunogenicity, so causing immune reaction, is a lot lower because you're giving this drug once, hopefully not more times. That would be very unlucky if you continuously get bitten by the same snake. Or maybe at some stage natural selection. But I don't want to assume here. But yeah, no, so I think that is one of the main things to consider. I think also one thing that is a bit overlooked is going away from therapeutics. It does have industrial applications. So looking at agriculture, targeting agricultural pathogens, increasing food safety. So there's a lot of viruses that are ravaging through different crop plantations. Why not use some of these AI proteins to inhibit those and improve also our food safety? There's also industrial enzymes that need to only function in different conditions. You can use design proteins to work with that. And really the next big frontier is designing enzymes, so very different types of proteins completely from scratch. That's the next big thing I think we'll see in protein design as well.
Have you looked into AlphaFold 3 at all? If so, what are your thoughts on how this might integrate with or change your workflow or how you do things?
Yeah, no, we've looked at it from day one, basically. We were very excited, as most of the rest of the scientific world that is working in this space. I mean, it's obviously very annoying that right now we can only generate 20 proteins a day. It's a very closed project. It's going against what I like to do, which is very open science. But they are plans and they are going to open it up. They have promised. I'm pretty sure if they don't, somebody will just reverse engineer what they did. But going away from that, we like it a lot and we're very excited about it. The frustration just stems from us being so excited about it and we just want to use it more. And it will 100% directly click into our design pipeline because right now we're using AlphaFold 2 to evaluate. We've seen that AlphaFold 3 performs better in basically every case that we've at least investigated. And so we're probably just going to replace AlphaFold 2 with AlphaFold 3, and that will hopefully increase the success rate that we're seeing from our de novo design binders in terms of translating them into actual successes in the lab. So that's very exciting.
Right. So it's the sort of thing where you replace AlphaFold 2 with AlphaFold 3 and hypothetically your hit rate just goes up. That's basically it. There's no other contingencies to think about?
Not really. I mean, obviously, new tools, new problems, but we'll see about that when we get there. But we've tested it in small use cases already, but we can design some structures and it's shown a lot of promise there. And Google DeepMind also put out their own protein design tool, AlphaProteo, and that looks really exciting. And they've shown a pretty decent improvement over also RF diffusion. My intuition, it's hard to tell from the paper, but my intuition is that a decent chunk probably comes from also using AlphaFold 3 in the evaluation of their design binders. That their filtering is just going to be more stringent and better. So I'm excited to see how this actually plays out in a protein design space.
Let's shift gears a little. You're doing some exciting work around quantum computing. Can you tell us a little bit about that work?
Yeah, happily. I mean, I got basically conned into this, I have to say. Up until a year ago, so I would have told anybody who asked that quantum computing is witchcraft and it's completely nonsensical to spend time on this. But then I'd say two major things happened that changed my mind. One was my head of department said, "Hey, Tim, quantum computing is pretty cool right now. Check it out. You're head of data science. You need to inform yourself." I was like, okay, actually, he's got a point. I probably should at least be informed. So I'm not just complaining without any knowledge. So I started going to a couple of presentations and so on and started being interesting, but still couldn't really see how this applied to any of our use cases. But then I'm also part of what is known as YATSE, great name, I know. It's the Young Academy of Technical Sciences and Innovation here in Denmark. This academy brings together a bunch of young engineers, scientists from different areas within the sciences, both from industry and academia. And one of the people I met there, Sophie, she was the quantum business developer at company called Sparrow Quantum. We had a great chat over dinner and she told me, yeah, that quantum is really cool and I should really look into it. And they'd seen some success with quantum combined with machine learning in image AI. And that really caught my interest because once I heard that quantum together with ML help with image AI tasks, this rang a bell because, as I mentioned before, image AI has been playing a big role in what we're doing in protein sciences. So I started reading up a bit more about the topic, and I started getting pretty excited. I involved one of my PhD students, Jonathan Funk. He luckily was also very excited to play around in the quantum world, and we started a collaboration with Sparrow Quantum, Orca, which actually makes quantum computers as well, and looked at how can we use quantum computing to design and generate peptides. Now, peptides basically being small proteins, and the reason why we're going for these things is because quantum computers still aren't super powerful, and we need to focus on less complex problems. Eventually, we want to go into proteins, but right now, peptides. And the interesting problem in the, I'd say, therapeutic world is vaccines. So far, we've talked very much about antibodies and antibody-like treatments with these designed binding proteins, but vaccines is the other way around, where you just say, hey, immune system, this is your target, make sure you take care of this target. You're basically just forcing it to look at the evil enemy. And so there's two, I'd say, key things you need to do well to make a good vaccine. There's a lot more, but let's focus on two. One is you need to show it the right thing. So in the case of COVID, we had the spike protein and said, here, immune system, see spike protein, make antibodies so you can defend yourself. The other thing is the way these vaccines work is you give, especially if we're talking about mRNA vaccines, you insert this protein into the cell, and the cell actually, the information of this protein gets inserted into the cell. And the cell produces this protein inside. It gets chopped up and then presented as a peptide on its surface. But to actually be presented, it needs to bind to that receptor really well, which is known as being immunogenic, so actually presentable to the immune system. So you want the right peptide and you want one that is immunogenic. And we focused on the first part of this problem where we trained a quantum-informed generative adversarial network, a QGAN, to see if we could get it to generate peptides that were immunogenic. And we compared it to a classical generative adversarial network, same input data, same training regime. And we found that not only the quantum approach created more immunogenic peptides, but also was extremely fast at learning it. So even after one epoch of learning it, was very, very good at generating these peptides. And this is very recent data, so there's a lot of digging we have to do, a lot of cross-checking. We need to try a lot of different things and dig deep into the data. But as a first, I'd say, result, this was very, very exciting. And not being from the quantum community, but having presented to the quantum community at a conference called Biotech X last week, it seems like the quantum community is also reasonably excited because there's not that many use cases and not many successes of quantum actually showing superiority over classical approaches. So we've got a lot of efforts now digging deeper and trying to maybe make our first proof of concept quantum vaccine. Let's see.
Do you have any hypotheses about why quantum computers might be better at this type of algorithm than the processing units we have on our machines today?
Yeah, so the beauty here is we're letting both the GPUs and the quantum computer do what they are very good at doing. So the main thing that we did here is not live quantum machine learning. What we did is got the quantum computer to generate a quantum distribution. So the way that these generative adversarial networks work is basically you have a generator that generates peptides and a discriminator that tries to catch it out and say, nah, that's not real, that's not real. And it's fed input data, right? And the other thing that goes in is a distribution, which is basically a map. This is the playground you have. This is the box of Legos you get to build your tower. And we decided to change this playground, change this Lego box from a classical Gaussian distribution to a quantum distribution. And our hypothesis was, you've got a different set of bricks. Maybe you'll build a different tower. Maybe that tower will be taller or prettier or nicer, or at least different. And so that was the main hypothesis here, is that we are giving it a different framework to explore within. And what we wanted to see is something different. We were hoping for something better, but at least something different. And the cool thing is when we actually looked at the way that this quantum-informed GAN was learning, it was actually generating peptides that were more diverse. It was exploring a lot broader than the classical one. And then we were just very happy to see that that also translated to being quicker at generating things that were actually better and more useful. So that was the main hypothesis here.
Super interesting. Well, obviously, Tim, you've got some very fascinating research interests and some interesting successes to share. And it's been, a delight to have this conversation with you today. As we head toward the end, can you share where people can go to learn more about you and your work?
Of course, you can find my name, using Google, I guess. But just Google me on the DTU website. Very active on LinkedIn. Feel free to reach out there. We've also got a website called digitalbiotechnology.com, digital-biotechnology.com. And feel free to check us out and reach out if you're interested in anything we're doing or want to collaborate or want to hear more. Always happy to chat. And I have to say it was an absolute pleasure also talking to you this evening, Ross.
Yeah. Well, thank you, Tim. Really appreciate the time today and look forward to connecting down the line.
Same here.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.